Why, hello.
Welcome back, everyone. This is the Basis Middle School Use of Book Club. Today we'll be covering Campbell's chapters 44 through 47. So, kidneys, hormones, reproduction, and development. A lot of physiology today. Let's get into it. Chapter 44, we start off with osmoregulation and excretion. Those are two functions of our kidneys, and let's talk a bit about them. So, osmoregulation is controlling the concentration of solutes in your body. So keeping your sodium, potassium, calcium, the levels of different ions and different all kinds of solutes within your blood and your interstitial fluid. You're maintaining a certain ratio between them, right? And animals can either be osmoconformers or osmoregulators. Now, an osmoconformer takes up the concentration of the environment, and what that means is in imagine if a fish in water will have an osmoconforming fish will have the same interstitial fluid concentration of sodium as the water around it. It's like an animal that doesn't control its body temperature. Um, Osmoregulators are it's the well, they regulate their osmolarity. They control their solute levels regardless of the environment, right? As you can see here, one method of regulating osmolarity is determined by how much urine you produce, right? If freshwater fish gain a lot of water through osmosis, because their environment is hypotonic to their cells will continually uptake water from the environment, and that dilutes their blood and their interstitial fluid. So they need to excrete a lot of water as urine, um, through urine. Um, Marine fish, on the other hand, their environment contains a very high concentration of salt. So water leaves their body through osmosis. Um, and they take in a lot of salt. So they need to put a lot of salt into their urine so that their interstitial fluid doesn't get oversaturated with these ions, right? Um, and transport transport epithelia. These are this is a it's just a vocab term. It's on the slide because with it's generally associated with nephrons, kidneys, and this is what how your body puts things into the urine and takes things out of the urine, right? These are generally what's it called? Simple or stratified cuboidal. So that means that the cells are like cubish in shape, and they're either a single layer of cells or like usually two layers of cuboidal cells, right? And yeah, this is how you put solutes or things you want to get rid of into the filtrate. The filtrate is another term for the urine. Okay. And one major um one solute that all animals need to control is their nitrogen, these amino groups, because if all proteins have every single amino acid has an amino group. That's part of the name. Um, and all nucleic acids, because they have nitrogenous bases, they also contain they also contain nitrogen. So you're as we live and breathe, we work with nitrogen a lot. And when metabolizing these different macromolecules, we end up with a lot of like nitrogenous waste. And if that builds up, that is harmful to all living beings. So how do the way that animals compensate for this amount of nitrogen is they get rid of it, but how they get rid of it is different, right? Some animals excrete it just as it is, as ammonia. They don't do anything to it, and the this is, I believe, more dangerous. But it's not. You don't need to spend any energy, and you don't need to spend any energy to condense it. Unlike uric acid here, where you spend energy to condense these four nitrogens into one compound, and that makes it easier to get rid of, but it's also more costly. So there's this balance between this ammonia that's harder to get rid of, but less expensive to get rid of, and the uric acid that is easier to get rid of, but it's much harder. It's much more costly to produce. And we mammals, we're mammals, we produce an intermediate called urea, which is not as it's you're kind of condensing it, but not as much, not to the level of uric acid, right? Yeah, most aquatic animals excrete their nitrogen as ammonia. Mammals and adult amphibians convert that ammonia to urea, and then these animals convert ammonia to uric acid. One maybe trivia thing is uric acid. Well, I think it is guano. Uric acid is no, actually no, I got it wrong. Sorry. Nitrogenous waste. Okay. Now let's talk about the filtration units of kidneys or excretory organs. How do these excretory organs do their jobs? So let's just dissect this one part at a time. So your kidneys filter your blood, right? They take a bunch of things out of your blood, and then so first you have blood that runs through this Bowman's capsule, right? They run through this capillary network called the glomerulus. And one like vocab term is this glomerulus is fenestrated, which means that it has large holes that allow a lot of liquid to flow out of the capillaries. That the hydrostatic pressure pushes the water out of the capillaries and into the proximal tubule. And this is now the fluid is called filtrate because it's been filtered out of the blood, right? An important thing to note is that like large proteins and cells, blood cells, platelets, they're filtered so that they can't enter the filtrate normally. They can't enter the nephron. They can't enter the proximal tubule. Yeah, and then the liquid snakes its way, the filtrate snakes its way through the proximal tubule, down through the loop of Henle, then up through the distal tubule, and the final product is released into the collecting duct, which branches with other collecting ducts and empties into the bladder. And along the way, you might think, why doesn't the filtrate just go straight from here to into the duct? Why do you need to go through this entire long process? And the answer is that when you're filtering, the filtering step in the glomerulus, it does filter out proteins and blood cells, but there are a lot of, you're basically taking out the liquid portion of your blood. And there's a lot of valuable things in there that you don't want to get rid of. Like, for example, like water. If you don't reabsorb any water from your filtrate, you die. There's no way, you can't replenish it. I'll get into this later. Another thing is that you want to reabsorb a lot of ions that are leaving your blood. You want to reabsorb sugars, you want to reabsorb anything that's in the filtrate that could be valuable, right? And then this is a simplified version of how the kidney works. So blood enters into this is a capillary bed. I don't know why they're calling this a capillary, but blood enters into this capsule, and then what fluid is pushed into the excretory tubule, and then as you can see here, things are exchanged between the blood and the filtrate as required. Yeah. I guess this is talking about the path. It's not in any particular order. You should know that the collecting ducts empty into renal pelvi, and then the renal pelvis, the renal pelvis empties into the urinary bladder, and then it leaves through the ureter. Urine leaves through the ureter. Just making sure I covered everything. I think kidneys are so cool. I love kidneys. Absorption, secretion, excrete. Yeah, I'll just cover these 4 definitions first. Filtration, that's just pushing out fluid and then modifying that fluid through the kidneys, right? Reabsorption and secretion are different. That's how you modify the filtrate. Reabsorption is you're taking back valuable things. Secretion is you're putting in garbage, right? So yeah. Okay, I'll just explain. So when your blood runs through the glomerulus, not everything in the blood gets processed as filtrate, right? There's a limit to how much fluid can go out of the capillaries, and some of the waste remains in the blood, and as a result, you need to actively secrete that waste back into the nephrons, so that they end up in the urine and out of your body. And the excretion is the final, like, you're peeing. Yeah, you're just getting rid of everything you don't want. Riva?
I have a feeling someone was watching YouTube.
I think I might know who that is. Let's pay attention, please. Come on. The nephron is organized for a stepwise processing of blood filtrate. All right, so let's go. Oh, okay. That's fine. This is a diagram. No, you're fine. This is a diagram of the structure of a kidney, right? Oh, sorry, of a nephron. What am I? This is the structure of a nephron. So this is a proximal tubule. Filtrate goes in here. It's modified in the proximal tubule, down the loop of Henle, through the distal tubule and down the collecting duct. All right, so let's talk about the proximal tubule first. And you know what, I can explain this. So one really cool thing that I found was that was how the proximal tubule recovers salt and water, right? So what they do is they pump specifically just sodium into the cells of the proximal tubule. So this tubule, think of it as you have cells lining it's a tunnel, and the cells that line there are cells that are lining a tunnel, right? And you're pumping sodium into those cells. And then as a result, it creates an electrochemical gradient because sodium is positive and chlorine is negative, right? So all the chlorine starts passively diffusing in towards the sodium, and that forms sodium chloride, right? And because there's a higher concentration of sodium chloride outside than inside, water will flow by osmosis outwards. Isn't that they just by pumping salt, they get chlorine to follow and then water to follow as well. I think that's super interesting. I don't you probably don't need to know that for BioVolt, but let me know if you want me to reiterate that. Yeah, so in the proximal tubule, you reabsorb sodium chloride, potassium, water, nutrients, and some of these nutrients include like glucose or just anything in your blood that could be of use. You can also take out bicarb or put in hydrogen. So in this way, you can control the pH of your blood. So if your blood pH is too low, you put hydrogen into the filtrate and your pH goes up. If your pH is too high, you take out bicarb from the filtrate and then your pH goes back. No, wait. If your pH is too high, you take out bicarb. Actually, I'm not sure how the bicarb thing works anymore, but the proximal tubule regulates pH in some ways, and so does the distal tubule. Does that make sense? So you reabsorb in the proximal tubule, you reabsorb a bunch of things and then control pH. You also excrete ammonia here. All right. So now when we get to the, sorry, hold on. I'm gonna go grab some tissue. Give me 1 second. Sorry about that. I wanted to make sure that I could talk about the loop of Henle well, because this is a doozy. So, now you get to this really awkward looking structure, right? Wouldn't it be so clean if this just went straight from here across down and then down, right? But no, there has to be this loop reaching deep into the kidney. And there's a good reason for this. The loop of Henle reabsorbs water and salt. So it takes out water and salts from your filtrate. And how it does this is it creates a gradient of concentrations. It relies on something called the countercurrent multiplier, which I'm not gonna go into the details now, but if you're interested, you can look that up. I found a good video on it. Yeah. So basically you put the thick segment of the ascending limb. All right. Let's get this clear. So you have the descending limb. That's just one part, one descending limb. And then you have a thin segment of ascending limb, and then you have a thick segment of ascending limb. So one descending limb, and then the ascending limb is split into two parts, right? And the ascending limb will pump out salts. So the ascending limb pumps out a bunch of salt into the environment. And this salt kind of settles lower so that the inner medulla of the kidney is much more concentrated than the outer medulla and the cortex. So what happens is as you go down, the concentration of salt and urea increases ultimately by a factor of 4. So the like right here at the bottommost tip of the branch, there's 4 times as much salt and urea as near the top. And this is important because let's say you have a filtrate, your filtrate is moving down, and as it's moving down, it starts to encounter a hypertonic environment, right? The outside has more salt than the inside. And so it will lose a bit of water. But as it keeps going down, even though it's losing water and becoming more concentrated, the outside is also becoming more concentrated. So it keeps on losing water. It keeps on losing water all the way down until it gets to this point, where as it goes up, now it's less concentrated than the outside. And through ion channels, salt will leave the inside. But as it's moving up, even though it's losing salt and becoming less concentrated, the outside is also becoming less concentrated. So the salt will keep leaving, and then until it gets to a certain point where the salt won't leave passively, and you have to pump salt outwards. Right. And then that's what the ascending limb does. So the ascending limb will pump out salt to maintain this gradient. And the result is that you take away a bunch of water and salt from the filtrate, and then you're just reducing the overall volume of the filtrate. So let me repeat this. As you go down, the outside of the kidney becomes more concentrated in solutes, right? And as the filtrate goes down, it starts losing water. But even though it's losing water, because the outside is getting more concentrated, it keeps losing water until at the bottom it's very, very concentrated in salts. Then as it goes up, it starts losing the salt. It starts losing the salt. But because the outer environment is getting less and less concentrated because you're going up, you keep on losing salt passively until you get to the point of the thick segment where you have to pump out salt actively, right? And then you keep going. Then you get to the distal tubule, and the distal tubule, there's a lot of active reabsorption of different solutes. You can pump in potassium and hydrogen. Here you can also control the pH and blood potassium levels. And then once you get down, the filtrate, once it's finished being processed, it gets to the collecting duct, and what do you know? It's actually not finished process. It's actually not finished being processing. And then as it moves down, salt will leave, water leaves, and urea leaves. Right. Yeah. Okay. Let's talk about some of the hormonal circuits that link kidney function. Let's talk about some of the hormonal control of the kidney. So the posterior pituitary releases a hormone called ADH, or antidiuretic hormone, right? And diuresis is peeing a lot, so releasing a lot of water. So antidiuresis is you're peeing less. And what that means is that when you have ADH in your blood, you're going to be recovering a lot of water from your filtrate. And specifically, this takes place in the collecting duct, right? As you remember here, you're getting water back from the filtrate, and ADH will act on here. So the more ADH you have, the more water, the more receptors you get, and then the more water that you pump back into your system. And then let's talk about the juxtaglomerular apparatus. It runs two systems. Actually, I think it only runs one, but ANP also relates to kidneys, and we'll talk about that later. But it when it detects that there's not enough, or when there's not enough flow in the kidneys, it will actually no. No. When it detects that when it detects that there's not enough flow in the kidneys, it will secrete something called renin, which gets changed to angiotensin. Angiotensin 1 goes to the lungs and becomes angiotensin 2, right? And then angiotensin 2 goes back to the adrenals and then causes the adrenals to produce aldosterone. Aldosterone will cause sodium and water to be more to be re more sodium and water to be reabsorbed, and that will bring up the blood pressure and volume. So if there's low flow through the kidneys, that means that there's not enough blood volume, right? Remember that the amount of filtrate depends on how hard the water is being pushed through the glomerulus. So if there's not enough blood pressure and blood volume, that means that there's not enough filtrate being pushed out, which tells the JGA you need more sodium, water, and blood pressure. And then ANP is like the opposite effect. So angiotensin and aldosterone make your blood pressure go up, right? ANP will decrease your blood pressure. So atrial natriuretic peptide is a hormone that's produced actually by your heart, by your atria, hence the name. It is sorry. It's released by your atria when your blood pressure is so high that it like stretches the walls of your atrium, right? When your atria stretch, that you have too much blood. You have too much blood volume. So as a result, your atrium secretes ANP, which causes your kidneys to excrete more urine, more water and salts in your urine, and decreases your blood pressure. And as a result, your artery, I keep messing up arteries and atria. Your atria expand less, it secretes less ANP, and then it's a negative feedback loop that results in you getting normal blood pressure. I need to speed through the rest of the slides. We're like 8 out of 20, but we're halfway through with our time. So hormones, types of signaling, paracrine, autocrine, and endocrine, right? Paracrine means you're secreting a hormone that acts near you. Autocrine, you're secreting a hormone that acts on yourself. Endocrine, you're secreting a hormone that moves through the blood. Oh, I'm going to clarify this in 1 second. You're secreting a hormone that goes through the blood and moves out all over the place, right? It can, like, a epinephrine produced in your adrenal medullas, which are on your kidneys, can go to your liver, to your heart, to your they go everywhere. That's endocrine. And I'll just talk about this real quick. A hormone, like strictly definitionally, is a molecule. It's a small molecule that's present in small amounts that is circulated through the body by the blood. And one important thing is that for autocrine signaling, you might think it's just like spitting it out a little, and then it attaches to its own receptors, right? No, that's not the case. Yeah, for example, your stomach secretes gastrin. That gastrin will go into the bloodstream and circulate around back to your stomach where it acts on. So all hormones are secreted into the blood. Classes of hormones. Hormones can either be gases like nitric oxide, polypeptides like calcitonin, steroids like your sex hormones like aldosterone, glucocorticoids, and amines like I almost say glucose, dopamine, epinephrine, norepinephrine. those are specifically called catecholamines, just for the record. And your endocrine system is regulated by, I say regulated by, they kind of regulate each other. So your hypothalamus regulates your pituitary regulates the rest of your body, and the rest of your body regulates your pituitary. So how do they do this? Well, okay, so let's talk about the classes of hormones that the hypothalamus and the pituitary hormone that the pituitary and the hypothalamus secrete. So the hypothalamus secretes tropic releasing hormones. Actually, let's talk about the pituitary first. The pituitary will release in stimulating or inhibiting hormones. Does it release inhibiting hormones? No, it does not. So the does it? Wait. I don't think it does. So the anterior pituitary will secrete what are called stimulating hormones, right? So for example, thyroid. Let's use the thyroid as an example. So the anterior pituitary will secrete a hormone called thyroid stimulating hormone, and stimulating hormones have two effects. One, it causes that hormonal gland, that endocrine organ to grow. So if you have a lot of TSH, your thyroid will grow big, and that's never mind. If you have a lot of TSH, your thyroid will grow big. The second effect is that you'll produce a lot of the hormone of that gland. And the hypothalamus regulates the pituitary by secreting what are called tropic regulating hormones. Tropic regulating hormones. So these stimulating hormones are called tropic hormones because they cause the endocrine glands to grow. The hypothalamus, for example, will secrete thyrotropin releasing hormone, which basically tells the pituitary to release thyroid stimulating hormone. So thyrotropin releasing hormone, thyrotropin releasing, thyrotropin is TSH, thyroid stimulating hormone. Releasing hormone is it tells the pituitary to release that hormone, right? So let's go adrenocorticotropic releasing hormone. What does that do? It tells your pituitary to release adrenocorticotropic hormone, ACTH. Does that make sense? I feel like I went through that really fast, and this is kind of a confusing topic. Let me know if it does. We're a bit short on time. You can stay after and ask me. Yeah. More hormones, thyroid hormones. Thyroid hormones consists of T3 and T4. So triiodine thyroxine or tetraiodine thyroxine, right? And these act with so two things you need to know. T3 is the metabolically active one. So T4 is present in the blood, but at lower constant. T4 is actually this is a bit fuzzy, but T3 is the active one, and that's the one that actually does a hormonal function. T4 can be converted into T3, but T4 itself has no effect. Thyroid hormones will work with vitamin D. So thyroid hormone works with vitamin D to up your metabolism. It's a bit strange, but I don't know exactly why. That's just how it works. I'm sorry, I can't give a more satisfactory explanation. Your parathyroid gland is also very important, right? Your parathyroid secretes a hormone. It's literally called parathyroid hormone. And what this does is it raises your blood calcium level. This is very important. I don't have any specific examples I can just say that calcium is important for like almost all cells, like even for like signaling pathways. It's very crucial. back in the day, they didn't know the parathyroid existed. And so when they did like thyroid, when they tried to cut out someone's like part of their thyroid, they would just die. And then they didn't know why. And it was because of the parathyroid. Don't quote me on that, but yeah, don't quote me on that. Yeah, okay. And then the adrenal hormones, these hormones, um, the adrenals sit on top of your kidneys, right? This is an endocrine gland on top of your kidneys, as you see here, and it's split into two parts. So you have the cortex, which is like the outer part of your of the adrenals, and they have the medulla, which is the inner part. And the medulla is easier, so let's talk about that first. The medulla secretes catecholamines. It secretes epinephrine and norepinephrine, and these two will they initiate the fight or flight effect. Um, they, yeah, I mean, yeah, they initiate the fight or flight effect, and yeah. Now, and then the adrenal cortex, the cortex is split into three layers. You don't need to know what the three layers are, but maybe know that it consists of three layers. And it produces a lot of different steroid hormones. So like glucocorticoids, mineralocorticoids, um, a little bit of your androgens, like your sex hormones, some estradiols, yada yada yada. One interesting thing like about glucocorticoids, we already talked about mineralocorticoids. That was like aldosterone. It makes your body take up more water and minerals, right? But glucocorticoids, they cause your body to break down fats and release glucose in your bloodstream, and it also suppresses your immune system. Um, yeah, I guess that's some interesting trivia. Glucocorticoids suppress your immune system. Um, Sex hormones secreted by your sex organs. Uh, I mean, yeah, they cause yeah. What's up?
What is norepinephrine?
Norepinephrine is I mean, so it's a hormone and a neurotransmitter. I don't know. I don't know like the actual molecular um structure of it. I know it's a catecholamine. It's like a ring with a nitrogen in it. Um,
Okay.
so it works.
What are
It's
oh, I'm sorry.
go ahead. Go ahead.
What are corticoids
Oh,
again?
mineralocorticoids. So these two. Um,
Yeah.
so mineralocorticoids, these are like steroid hormones that mineralocorticoids caught. Like we talked about them before. These are the aldosterones, right? They they make you take up water and salts from your bloodstream.
Yeah.
Um, yeah. So retention of water, sodium ions in water, increased blood volume and blood pressure, and glucocorticoids, they kind of
Increased blood glucose.
yeah. So increased blood glucose, you break down proteins and fats, and you suppress your immune system. Which an interesting thing is that this axis is activated by stress. So if you chronic stress will lead to excess secretion of glucocorticoids, which will suppress your immune system, making it more easy for you to get sick.
Oh, is
So
that what?
yeah. Cool.
Sorry. Is that why like um people always tell you to not like stress out too much, otherwise you might get sick?
I can only say that I'm not a I'm not a doctor. I can only say that it's probably a contributor. Um,
Okay.
yeah, it's a contributor. It's not the only thing, but yeah. Okay, were you good? Oh, okay. Anyways, I'm move on. Animal reproduction. So there, we reproduce sexually, but a lot of animals don't agree. They reproduce asexually, and here are some examples. Um, okay, never mind. The first row is all asexual reproduction. So budding, a new organism grows on you. So imagine like you grew another arm, but that arm becomes your child, right? It like pinches off and becomes a child. Like that's basically budding. Fission is, imagine you grow big and then you split yourself. It's like cytokinesis and the cell cycle, except you're creating a new individual. So it's reproduction,
Mm.
right? Fragmentation is you have your one organism can like split itself, like a large organism can like split itself in like pieces, and each pieces, if they grow back to an adult, that is reproduction. So like for example, some planarians, you can cut them. These are like entire worms. You can cut them straight down in half and they can regenerate their entire bodies. A better example, maybe starfish. You can cut off an arm, and if that arm grows back into a full individual, then it's reproduction. And parthenogenesis, um, generally, if an animal has an egg, you need to activate that egg to like have it begin its development. So usually the activator is sperm, but in case of parthenogenesis, the population is all females. There's no sperm involved. The eggs just activate spontaneously and begin development. Hermaphrodites, these are sexually reproducing organisms, but they have both sets of sex organs. Yeah, well, some like there are snails, earthworms. They cross fertilize each other, when they reproduce. And then sex reversal. This is when a member of a population will change their sex to like suit needs. So, like for example, I think I forgot to look this up, but I think it was some kind of parrotfish. The largest member of like a tribe, not a tribe, of like a a a mating group, I guess. The largest member of a mating group is the male. If that male dies, the largest female becomes the new male. So, yeah, yeah, sex reversal, you change sex. Fertilization, external versus internal fertilization. So fertilization marks the process where sperm enters an egg and initiates its development, right? And this can happen either externally. So frogs will introduce their gametes into their surroundings, and they like swim all over the place and then randomly find each other. Internal fertilization, well, we have internal fertilization, use your imagination. But external fertilization, one interesting thing is that for external fertilization, generally you're releasing your gametes into water where other species also release their gametes. So you need stronger barriers to your female gametes need stronger barriers to entry from sperm of other species. And generally frogs have certain blocks to polyspermy that humans don't have. Yeah, we'll get into that in a bit, but yeah. Also external, you generally secrete more gametes. Like humans make one female gamete a month. Frogs just go crazy. All right, the male. Testes, accessory glands. All right, let's just get into this. We don't have time. So sperm are produced in the testes, and then they start off near the walls of the testes. They move inwards as they develop, and then they go through the epididymis to mature. So sperm start off as germ cells at the edge, then they migrate inwards and start developing, and then they go through the epididymis to finish their development, right? And during ejaculation, they get squeezed through the vas deferens and combined with fluids from these accessory glands. So there are three in males. There's the seminal vesicle, the prostate gland, and the bulbal urethral gland, right? Uh, I don't remember the distinction of the fluids secreted by prostate and seminal. I know that bulbal urethra will secrete things that neutralize the any possible remaining urine in the urethra. Um, because as you may or may not know, urine is slightly acidic. So the bulbal urethral gland will secrete things that neutralize that acid. The seminal vesicles in the prostate gland, I guess one thing that you should know is they secrete ascorbic acid, which is vitamin C. They also secrete fructose, which is the primary food source of sperm. They secrete both coagulants and anticoagulants. Um, uh, oh, Marco, by the way, you're unmuted.
Sorry.
Oh no, you're fine, you're fine. Um, I think that's it. I think that's it. Yeah. Also, I can talk about this. So in your testes, there are two types of like specialized cells. One are called Sertoli cells, and these like nourish and care for the developing sperm. The other are Leydig cells, and Leydig cells produce hormones. It's like sex hormones, you know, the female ovaries, ovida. Why is this so small? Um, Here. If I do, oh, my bad. I, oh, my bad, my bad, my bad. All right, okay. So, let's work our way in, I guess. So you start off with the outer opening. You have the labia minora and majora. These are like fatty folds of skin. You have the vaginal opening, the vagina, the cervix, just like the outermost part of the uterus. You have the uterus itself, then the ovary and the oviduct. You can get a better look down here later. And then here the clitoris is like a I forgot, it's not a homologous structure. It's similar in origin to the penis, where it has the body glands, it has its erectile tissue, and if this person was a male, then the clitoris would have developed into the penis, right? Oh, hold on. Let me zoom out. Here you can see how the ovary is connected to the uterus through both a ligament and an oviduct. Uterine wall, endometrium. Yeah. Gametogenesis. Let's talk about this for a bit. Hormonally regulated. The anterior pituitary secretes follicle stimulating hormone and luteinizing hormone. So we talked about Sertoli and Leydig cells. Sertoli cells are stimulated by FSH. Leydig cells are stimulated by luteinizing hormone. Care for sperm produce sex hormones. In the females, granulosa and theca cells, they both care for the developing. Excuse me. They both care for the developing female gamete, but the granulosa cells secrete androgens, and the theca cells the granulosa secretes estradiols, which are female sex hormones, and the theca cells secrete male sex hormones. It could be the other way around, but one of them, each one secretes one type of sex hormone. And but they both care for the female gamete. And then the ovarian and menstrual cycles. So the menstrual cycles, the menstrual cycle talks about or pertains to the cycling of the endometrium, which is like the inner lining of the uterus. So like when it grows, when it thickens, why did polar bodies even exist? Um, you know what? I can't give you 100% guarantee sure answer. I would say that it's because female gametes you need there are a lot of things in female gametes that are very important. Like sperm, you just drop a nucleus and that's it. You're done. That your job is done. But for eggs, you need to set up a bunch of things like effect factors. You need to you need to care for the egg. The egg needs to be able to be big enough to split into a bunch of cells. And therefore, you need a lot of cytoplasm in the final gamete. And I guess that's why polar bodies exist, is because you need to like funnel all of your cytoplasm. Yeah, okay. And then the ovarian cycles measure like the release of different of female gametes of like it's the cycle of things that happen in the ovary. Yeah. And now we get to development. This is what I was talking about before: fast block versus slow block topolospermy. So in frog, let's talk about specifically frogs, right? Frog eggs, their plasma membrane has a bunch of receptors for sperm heads, and when the sperm head attaches to a receptor, it attaches, and then the receptor kind of pulls in the the sperm head slash nucleus, right? When that happens, the membrane will depolarize, and then all of the receptors will fall off. So that way you can ensure that there's only one sperm per egg. And why is that important? Well, if you have one egg and if you have one haploid egg and two sperm fertilize it at the same time, now you have a triploid individual. And that triploid individual, it's polyploidy, it's almost certainly going to die. So you don't want that. You want your offspring to survive. And as a result, you produce these fast blocks to polyspermy, blocks to many sperm, hence polyspermy. However, sometimes the fast block is enough. Like sometimes sperm can like force their way into the egg without the receptor. And as a result, there's a second block called the slow block to polyspermy, where the egg will release a bunch of like they're called cortical granules that are filled with calcium, and that calcium forms like a you know how calcium forms your bones. It's very strong. It forms like a shell around the egg, like a hard layer that it physically prevents sperm from getting in. So that's the slow block. It's called the slow block because it takes a while for all the calcium to release and stuff. Yeah. All right, cleavage. Cleavage is a repeated cell division of one more thing. Humans don't have a fast block. They only have a slow block. And that's one of the things I was talking about before, is that frogs release their gametes they have external fertilization. They release their gametes into their surroundings, and as a result, they need more blocks to this kind of stuff. Cleavage, blastula is formed. So basically, when a zygote is first fertilized, the egg will undergo a process called cleavage, which is repeated cell division without increasing cytoplasmic mass. So you're producing a lot of DNA, and you're just dividing repeatedly until you form something called the blastula. And the blastula is a hollow ball of cells. So you have like a bunch of tiny cells lining the outside, and then like a hollow bit in the middle. It's filled with like liquid, but not actual cells. In frogs, there's a distinction of the vegetal versus the animal pole. And the vegetal pole, frog eggs have some yolk, right? They have they have yolk, and as a result, one side has more yolk than the other. That the side that has more yolk is called the vegetal pole. Vegetal pole has more yolk. Got it? The animal pole is just the opposite side, has less yolk. Now let's talk about cleavage can either be holoblastic or meriblastic. So holoblastic means that the furrow goes all the way through the egg, right? It cuts the egg completely in half. But meriblastic, on the other hand, which is what frogs have, is you don't cut all the way, or it's slower on one side than the other. And the reason that it's slower is that the yolk in the vegetal pole slows down cell division. So it makes it harder for the cells like pull apart because it's thick and sticky. So you get this asymmetrical blastula where you have large cells at the bottom, small cells at the top, and this and the blastocele is near the top, right? Gastrula, germ layer. We talked about germ layers before. It's fine. Diploblast for the triple blast. It's in the name. Yeah. So I'm just gonna go. We have one more slide after this, which is on some pretty niche stuff. I'm gonna cover the amniotic egg just really quickly, maybe like half a minute, and then you guys are free to go. All right. So let's talk about the amniotic egg. We are amniotes, I think. Wait, no, yes, we are definitely amniotes. And the amniotic egg is characterized by four parts, right? So you have the amnion, hence the name, which cushions the embryo, right? And the amniotic sac cavity is filled with amniotic fluid, which has it cushions the embryo. Then you have the allantoid and the chorion, and both of these they help with gas exchange and waste removal. So allantoid and chorion help with gas exchange, waste removal. And the yolk is yolk. Well, it's like the food source, proteins, sugars, yada for the growing embryo. Okay. That's not everything, but like 99% of things. Honestly, if you guys want, I'll finish this, but you guys are free to go now. Thank you guys. So neurulation, this is organogenesis is like part of development where neurulation is the ectoderm folds inwards and you see here, it like pinches inwards and forms a tube. And it folds over at the top, and then it forms a neural tube. So this neural tube is called the notochord, and it will eventually form your spinal cord and stuff. Cell migration, not much to talk about. Apoptosis, that you just know that apoptosis is important in development. One crucial part is in your nervous system, actually. Most of the cells will die, kind of. Like most of your nerve cells will die. And this is called, actually, I don't remember what it's called, but it's not pruning. But it's a very important development because only the crucial neurons are left alive. So you're born with much more neurons than you actually should have. And the ones that are in the proper place will get nourishment and survive. The rest will die. All right. I'm just going to talk about determination versus differentiation because the other two are really not worth going into. Maybe cortical rotation, but no. So determination and differentiation, as you may know, differentiation is the process that a cell let's say you start with a stem cell, and the stem cell will differentiate into a muscle cell, right? It will choose to become a specialized cell. Determination is when a cell says, I'm choosing to become this cell. So think of it as determination is a muscle cell saying, I'm gonna sorry, determination is a stem cell saying, I'm going to choose to become a muscle cell. And there's nothing you can do about it, right? Once a cell is determined, you can't change its cell fate anymore. Once it's determined, even though right now it's still a stem cell, but because it's undergone determination, it will become a muscle cell no matter where else what you do to it. I get that's not entirely true, but it's mostly true. And then differentiation is the process that the stem cell actually becomes the specialized cell. So so determination might be receiving some cell signals. Differentiation is producing mRNAs, producing proteins, changing cell shape, yeah, stuff like that. Okay, well, thank you all for coming. I'm not gonna go over the last two because this is very niche. This requires a lot more explanation. Yeah, if you have any more questions, you guys can stay behind. I know I kind of had to rush through the material, but I really wanted to talk about Nephron. I don't know. Yeah. All right. Take care, Al. No problem. See you again. I'm gonna stay behind to do attendance. But
Okay,
yeah.
bye.
All right, bye, Isaac. Bye, Marco.
Thank you.
Yeah,
Bye.
no problem. Sally, you there? Nope, not. Well, yes, there. Okay.